Hello student, this is Dr. Faizan Mirza. In this video, I'm discussing with you some concepts regarding inherited change, and my discussion will focus mainly on mitosis and meiosis comparisons initially, and then I'll be moving on to how meiosis leads to variation. So let's start. Mitosis and meiosis, let's just recall we have sister chromatids present on one chromosome. This is the typical shape of a metaphase chromosome, whereas there are two sister chromatids having identical DNA, the same DNA, in fact, molecule, one DNA molecule and its copy present on the sister chromatids attached at a centromere. So this is the metaphase chromosome. So when we look at a diploid cell, we actually have a homologous pair of chromosomes. The homologous pair of chromosomes is a matching pair having the same number of genes. They have the same number of genes at the same gene loci. What is gene loci? Gene loci, gene locus, the location of a gene on a chromosome. So the gene present here will be the same as the gene present here. The gene present here will be the same as present here, but the allele can be different. Though homologous chromosomes, they both are of same length, they have the same position of the centromere, and the one of them is of maternal origin, one of them is a paternal origin. So you receive a homologous pair like this, one from your father, one from your mother. So one allele of this gene came from your father and one allele of this gene came from your mother. Both will form bivalent in prophase 1. Bivalent is when they pair up together during prophase 1. So when we are discussing the number of chromosomes and how they vary, how they actually go along when mitosis meiosis, let's just recall what mitosis and meiosis is. Mitosis and meiosis, in mitosis, there is a single division of the nucleus by prophase, metaphase, anaphase, telophase. And in meiosis, there's a double nuclear division, meiosis 1 and meiosis 2. Meiosis 1 will have all these stages, prophase, metaphase, anaphase, telophase 1, followed by cytokinesis. This then continues to meiosis 2, where again prophase, metaphase, anaphase, telophase occur, but now we'll add a Roman numeral 2, followed by cytokinesis. The number of chromosomes in mitosis, they remain same. The number of chromosomes in meiosis basically reduced by half. Homologous chromosomes do not associate here, but in meiosis, the homologous chromosome will associate to from bivalents. Bivalents are a pair of homologous chromosomes that form during prophase 1. Chiasmata are never formed in mitosis, but yes, chiasmata will form, crossing over will occur. Chiasmata will form, the chiasmata is a physical link between parts of homologous pair. Crossing over does not occur in mitosis. Crossing over will occur in meiosis. Crossing over means the exchange of genetic information between the non-sister chromatids of a bivalent. Daughter cells are identical. The daughter cells are not identical to parent cell. Two cells are produced. Four daughter cells are produced. The daughter cells have the same number of chromosomes. Daughter cells have the half the number of chromosomes as the parent cell. These are the basic facts. So how meiosis goes forward, we have discussed this in a detail in the same playlist in the video where I've covered details of steps of meiosis. You can go through this video if you want to recall what steps are there in meiosis. So I'm moving on to the concepts that how variation occur because of meiosis. So genetic recombination occurs in meiosis. What is genetic recombination? New variants are produced, recombinants are produced in the gametes by crossing over. So during prophase 1, the bivalents form. When bivalents form, this is how the bivalent form, the homologous pair. This is one homologous chromosome pair. The sister chromatids are there, and the non-sister chromatids they form a chiasmata, a point of breakage, and they exchange their segments. So this allow the chiasmata to form, followed by crossing over, where we can see exchange of equivalent segment occurred. So the alleles present here went there, and the alleles present here came here. So they exchange their segments. So the portion of chromosome they break off and rejoin. Combination of maternal and paternal alleles are produced. The recombination allows four different cells to be produced at the end of meiosis 2. If recombination doesn't occur, if it did not occur, then only two type of cell would have been produced. So how this goes about? This is what you can see here. This is one parental cell here having homologous chromosome where crossing over has occurred. Chiasmata was there. That's why it led to crossing over being occurring. In meiosis 1, the homologous chromosome gets separated. The homologous chromosome like moves away. One of them goes in one cell, the other comes in the other cell. The homologous chromosome separate. Meiosis 1 is called as the reduction division, where the number of chromosomes becomes half. Now, in meiosis 2, the homologous chromosomes, since they have already separated, now the sister chromatids will be pulled apart. Where the sister chromate is being pulled apart, this chromate goes on in one cell. This is the same paternal chromosome that was there in the cell here, and this one, which is a recombinant chromatin, it comes in the other cell. So this gamete is having a recombinant chromatin. This recombinant chromatin will have a combination of alleles of paternal and maternal origin both in the gamete. We are talking about the gametes of an adult. The adult is having the chromosome pair from his or her parents. So these are the gametes being produced in an adult. So this adult individual can end up producing a cell with a gamete having one chromosome, which is of his parent, exactly like his father, and another gamete which will have the allele combination of his or her mother, and then he or she will end up producing two more gametes, will have combinations equivalent of exchange segments of father and mother combination both. These are called the recombinant gametes. So this gamete and these four gametes are produced. So there are two parental class gamete and two recombinant gametes are formed. And these gametes will then go on and fuse to form the next generation. So they will fertilize uh, the gamete from the opposite gender. So, for example, if this combination goes, all the paternal alleles will go. If this is fertilizing, if this is fertilizing, then all the maternal alleles will go. If either of them is being used, the combinations will go on and pass on in the next generation after fertilization. So this is how it goes about. Now, let's move on. So, we are discussing the next thing here, and this next thing here is the random alignment of homologous chromosome. What is random alignment of chromosome? Homologous chromosome. The random alignment of homologous chromosome leads to independent assortment of homologous chromosomes, pairs, and therefore alleles in meiosis. How this occurs? Both maternal chromosomes move to one pole or both paternal chromosome move to the opposite pole. This is what's happening. In one case, this is case number one. In case one, we have both maternal and both paternal chromosomes moving to the poles in such a way that both maternal chromosome move to one pole and both paternal chromosome move to the opposite pole. How is this occurring? Let's discuss this first case. This is arrangement one. One paternal and one maternal chromosome having a tall stem allele and a short stem allele. This is having, for example, capital T. This is I'm showing the small t, the recessive allele, and terminal flowers. For example, recessive allele and the dominant allele of axial flower is there. So this cell belongs to an organism who is having a genotype of capital A, small a, capital T, small t, being heterozygous for the gene A and heterozygous for gene T as well. Now, this organism, when the homologous chromosome, they line up along the equator in such a way, what will happen? The chromosome will separate, the homologous chromosome separate. This chromosome goes up, so the T and A allele goes up, and the small t, capital A alleles will pull down because this is how the spindle will form. The spindle pulls them up, and the spindles pull these down. Now, when they have separated, eventually they will go by the end of telophase 1, they will go into separate cells. So by the end of telophase 1, a gamete will be produced having capital T with a small a, and another gamete will be produced with having a small t with a capital A. So there will be two type of cells forming at the end of meiosis if the arrangement was of this sort. This was the arrangement at metaphase 1. Now, what can be the second arrangement? The second arrangement goes that one maternal and one paternal chromosome move to one pole and one maternal paternal chromosome move to the opposite pole. In this way, the short stem allele chromosome and the terminal flower small a was like at the top and capital T, capital A chromosome were at the bottom. So this you can see is the difference. Now this because of this difference, when these chromosomes they get separated during meiosis one, once metaphase 1 has occurred, the random alignment of the chromosomes due the bivalent along the equator, they get pulled apart in such a way that the small t, small a allele chromosome goes to one end and capital T, capital A comes at the other end. This splits into two cells, one having both recessive alleles together, one having both dominant alleles together. This is how four different kind of gametes eventually are produced from an organism who was heterozygous dominant for two traits. what were the chances of a dominant allele coming with a dominant and another dominant allele? Both dominant alleles coming together, T and A, there was a equal chance, as equal as a small t, small a coming together. This was again as equal to the chance of small t and capital A coming together. And this was again as equal to the chance of small capital T, small a coming together. So in short, there was 1:1 chance of all these forming, or you can say 25% chance of all these type of gametes forming. This is called independent assortment of homologous pair and independent assortment of alleles. Four different type of gametes are produced, and because of these four type of gametes, they can have different combinations. So what have we discussed? We already have discussed that, for example, if an organism is having this combination heterozygous dominant, and you this is again, you cross it with another individual who is small capital A, small a, capital T, small t. So these both are dihybrid in this case. We know they both will produce four type of gametes. Their gametes will include the combination of all these four each. So we will have the offspring ratio 9331 forming. You have 9331 forming as the F2 generation. So F2 generation will be 9331. What is 9331? 9331 actually says that nine of them will have the phenotype dominant, and three will have the phenotype dominant and recessive, and the remaining three will have the phenotype recessive and dominant, and the last one will have the phenotype recessive. So this is how this will occur. So what are the combinations? You can work it out yourself and try to see what will be the four phenotypes here. The four phenotypes can be the length of stem and position of flower. So tall stem is dominant and axial flower is dominant. How many of phenotype will have tall stem and axial flower? Tall stem and axial means dominant for both. Nine will be dominant in the F2 generation. This is the nine. Then how many will be dominant recessive, capital T, small a? And this is what will be happening. Tall stem will be there, but the flowers will be terminal. So dominant and recessive, these will be three. That recessive dominant means that there will be short stem, but with axial flower. These will be three again. And lastly, how many will have the recessive combination, which means both recessive alleles together, one of those. I have discussed these ratios of 9331 in another video in the same playlist. You can go to that uh, for further discussion if you want. I hope this was helpful for you. That's it from my side. Thank you so much.